well, as you may know, I am Andrés Muro. I am working at the ICGEB already. This is something you know. I'm going to talk about a disease and a mouse model that we have developed in our laboratory that we are using just to understand Crigler-Najjar syndrome type 1 to get some insights into disease mechanism and therapy. So what is Crigler-Najjar? Crigler-Najjar is a disease that is related to the bilirubin pathway. I suppose that you all may know what is bilirubin. It's a compound that is a breakdown product of hemoglobin or from heme groups actually that are present in different molecules like hemoglobin or myoglobin or cytochromes. And then this heme is converted into biliverdin, then into bilirubin, and this happens in the liver. And then when bilirubin is normally an insoluble compound that is transported in the bloodstream bound to albumin, then it's discharged in the liver. In the liver, it has to be eliminated, and to do so, there is an enzyme that is called Ugt1 that conjugates glucuronic acid into the molecule of bilirubin, resulting in conjugated bilirubin. This conjugated bilirubin is soluble in water, and then this is eliminated into the bile fluid. When this enzyme is not working, then bilirubin accumulates, and then we have hyperbilirubinemia. Hyperbilirubinemia is a condition that could be physiological. It happens, I suppose, that many of you have children, and some of your children have to were in the neonatology unit under the blue light because of hyperbilirubinemia. This is a physiological problem, just is a delay in the expression of Ugt1, that is the enzyme, that happens just upon birth. In some cases, there are some complications and some other problems associated to the delay in the expression that could result in very high levels of hyperbilirubinemia that are toxic. And this happens without a mutation in the Ugt1 gene, and this is normally called neonatal hyperbilirubinemia, and could lead to kernicterus. I will explain you in the next slide what is this, and could lead to death. But there are other versions of hyperbilirubinemia that are caused by genetic mutations, and the one that we are going to deal with is Crigler-Najjar type 1, in which there is complete absence of the enzyme activity. Some other variants is type 2, in which you have just partial activity, but you still have hyperbilirubinemia, or Gilbert syndrome, in which the most common mutation are mutations in the promoter of the Ugt1 gene that reduce transcription and activity. So what normally happens is that, well, this disease is very rare. It hits more or less about one newborn per million. It is autosomal recessive, and it is very clear since the first days of life. So it has to be treated. If it is not treated, then could lead to very severe brain damage and eventually to death. And this is kernicterus, that is the coloration of the brain, the specific nuclei in the brain that lead to death when bilirubin levels are very high. If it is not lethal, then survivors may have very severe mental retardation or movement disorders, because it also affects the cerebellum, hearing loss, or complete deafness. So which are the therapies for this syndrome? The therapies available normally is phototherapy, as I mentioned before. Phototherapy consists in the irradiation of blue light about 450 nanometers, and that converts bilirubin into a photoisomer that is more soluble in water, and then this is readily excreted in the bile fluid. And as I mentioned before, children having this problem, they have to be under treatment all lifelong, and normally they have to be under the blue light for 10, 12, 14 hours per day, which affects completely the quality of life, the social life, and also the life expectancy. Because phototherapy, when the children grows up, then it's less efficient because the ratio between surface and volume is diminished, and also the skin gets thicker, and the efficiency of the light to get into the tissues is lower, and then less of this reaction is going on, and it's less efficient. So at the end of the day, what happens is that they have to undergo liver transplantation as a final therapy. So liver transplantation is a very invasive procedure, and if you look at statistics, these are numbers on patients that underwent liver transplantation of the Crigler-Najjar syndrome, and after 10 years, you have about 10%, between 5–10% of mortality, and about 20% of the transplanted patients had to undergo a new transplant because of organ rejection. So this is a very, let's say, difficult condition. So we are trying to understand better the mechanism of disease, and to try to provide new or alternative therapeutic options. Also for neonatal acute hyperbilirubinemia, one possibility when there is a peak of bilirubin that is not controlled by phototherapy, then what physicians do normally is an exchange transfusions. This is to replace the blood of the newborn by fresh blood without bilirubin, just to try to handle the acute situation of hyperbilirubinemia. So at that point, we constructed an animal model containing a mutation in one of the exons. Here is the scheme of the construct. You have here this yellow exon, that is the one that contains, we introduced a mutation here. I will show you the mutation then. We did a strategy using LOX-P sites and FRT sites just to eliminate the PGK cassette in vivo after mating our animals with a transgenic animal expressing the FLIP recombinase. And then we ended up with a construct that is similar to the wild-type construct, but contains the mutation in the exon 4. This mutation is a deletion that produces a frame shift, introducing a stop codon immediately after the deletion. That is very similar to what happens normally in patients. So patients have normally point mutation that change the reading frame and affect they don't get the full protein, and it is not active because at the C-terminal you have the transmembrane domain. This is a protein that is bound to the endoplasmic reticulum, and without the transmembrane domain, here is the point of the mutation, then the protein is soluble and gets degraded. Okay? So these are the animals, the mutant animals. You can easily see which ones are the mutant one and which ones are the wild type. This is a very good condition for us because you can see that the mutant animals are orange-yellow because of the high levels of bilirubin. And also it's very practical because at the very early days you don't need to genotype the animals, so you know that the animals are yellow, they are mutants, the other ones are wild type, and this is very useful for technical purposes. So I'm going to just try to explain you briefly what we have done just in this animal model to characterize the mechanism of disease and different type of treatments to understand also the mechanism of disease or to try to cope with long-term problems or with acute hyperbilirubinemia. So the first thing we did, as we have seen by northern blot analysis, is that we have less levels of the messenger, probably due to nonsense-mediated decay. And obviously, we have no protein by Western blot and no activity here. This is an analysis of microsomes from liver, and you can see here, this is the unconjugated peak of bilirubin after the HPLC, and this is the conjugated bilirubin that is absent, this peak in the mutant. And in the heterozygous, the activity is reduced. So you can see here, I don't know if you all can see, these are the bilirubin levels in the animals. This is the blood of the mutant animal that is practically yellow due to the huge amount of bilirubin that is present in the blood. And just one feature that makes the animal very similar to what happens in human is that this animal, if you don't treat it, animal die after a few days. So this is a survival curve. You can see here the wild type and the mutant, the survival of the mutant animals after 10 days. We have no survivor at all due to bilirubin toxicity. And this is what happens in kids when you don't treat them or you fail to treat them. So just you can see here that the cerebellum is severely affected. You can see here that the structure of the cerebellum is different in the mutant from the wild type. You have all the fissures that have a reduced shape, and also the layers of the cerebellum, the cell layers of the cerebellum are reduced. You have this specific coloration for Purkinje cells, in which you can see that there is a reduction in the number, there is a reduction in the dendritic arborization of the dendritic cells, and also the layer itself that you can see here that is well organized here, cells are very dispersed. This is an animal, these are two animals, wild type and mutant animals. You can see that the mobility of the mutant animal is highly reduced, and this is due to cerebellar deficits in which the animal has to open the legs to help himself to keep balance, and the movements are reduced. So this animal is about to die in 1 or 2 days after the movie because of these problems. So bilirubin raises quite a lot and produces brain damage. You can see here the discoloration of the brain. This is a mutant that is yellow, and this is the wild type brain. This is just a snapshot of the external appearance of the brain. So we did some different approaches to try to understand what happened with the disease. We did initially a proteomic approach in which we have seen that this is a proteomic approach of the cerebellum, in which the proteins affected were proteins involved in oxidoreductase or antioxidant activities. And we analyzed those proteins. Some of them we have seen that, well, in this case, the most damaged cells, this is a fluoro-JC coloration in which you can see neuronal degeneration. This is a specific coloration to see that. And the most affected cells are the Purkinje cells. You can see here, this is a coloration with calbindin that is in red, and this is in green you have fluoro-J. And you can see here the coloration with neo-N that does not stain does not stain Purkinje cells. It stains granule cells, and you can see here that very clearly that the most affected cells are the Purkinje cells. And the damage is related to the stage in which bilirubin peaks, and is related to the degree of development of each cerebellar lobule. We did also some analysis. We have seen that most of the proteins that were involved were related to P38 kinase, and this was highly activated. This is phospho P38 staining, also in the Purkinje cells. So having said that, we also did another analysis that is a transcriptomic analysis that you cannot see here, but it doesn't matter. Let's say we have seen that here the main downregulated genes are related to the cell cycle as cyclin D1, cyclin D2, and the main upregulated genes are related to transport and synapses. So we have seen that the cells this is a BRDU experiment in which we stain the cells with BRDU, and we have seen that after the peak of bilirubin that happens in P4, then you have less staining of BRDU, indicating that you have a reduction in the cell cycle. That is also related just to the stage in which the cells have to migrate into the cerebellum. So I will go fast in this part, and also this all the pathways that were involved were related to the IGF-1 pathway, in which we have seen that in the mutant animals also the local IGF production is reduced, and when we specifically treat the animals with a viral vector expressing the IGF-1 in situ, then we have a recovery in the mortality of the animal. So all these experiments lead to just to try to understand why the animals die, which are the mechanisms that are associated to bilirubin toxicity, with the idea of trying to find new therapeutic options just to modulate bilirubin toxicity. So regarding that, we analyzed also the treatment with phototherapy, try to understand which is the main window of phototherapy sensitivity. And this is the normal phototherapy unit that we are using in the laboratory, that is essentially the same to the one that is used in the clinics for the kids. So these tubes emit light of 450 nanometers, and they are exactly the same of the ones that you will find in the neonatal care units. So these Philips TL20 tubes. So we try to standardize the conditions with a meter, light meter, just to determine just the emission of the tubes and try to be always in the same conditions. So analyzing this, during our experiment, we transfer also the mutation into two different genetic backgrounds. One is the C57 black 6, that is the normal one that you have listened all over the course. And then we transfer also to another genetic background that is called FBB, just to see if there were some differences in the penetrance of the genotype in the different strains. And we have this, the answer was yes, because we have seen that the FBB background was less severe. It had lower levels of bilirubin. You can see at P2 and P4, the levels of bilirubin in the white strain, that is the FBB, they are lower. And also you can see here, this is the mortality curve of the C57 animals, and this is the mortality curve of the other animals, the FBB. You can see that it's delayed. They survive for more days. And when you treat them with phototherapy, then the black animals don't survive, but the white animals, they survive without any problems with phototherapy. So the phototherapy treatment is not efficient for the black animals unless we shave completely the animals every 2 days to avoid just the shadow of the fur into the skin. So also this strain of animals had severe problems in the cerebellum. You can see here that the different layers of the cerebellum they are affected, the external germinal layer, the Purkinje cell layers, or the internal germinal layer. They have differences in the depth of the layers. Also, they have differences in the number in the extension of the dendritic arborization in the Purkinje cell layer, you can see here this is a specific coloration with a calbinding antibody. This is the wild-type animal, and this is the mutant animal. And you can see how the treatment with the blue light fully rescues the phenotype. So we're trying to understand better which are the cell types and which are the window that is more affected. Then we did a treatment of the animals by treating the animals for different time lengths from birth to P8 or to P10 or to P15, and then looking at the survival of the animals. And you can imagine that the animals that received more blue light, they were less affected. That is quite intuitive, this. And this is what happened. So the animals that received light only up to P8, we had only 20% survival. The animals that received light up to P10, then we have about 50–60% survival. And the animals that received blue light up to P15, they all survive without any further problems. So this is what happens to the animals when the hyperbilirubinemia is very severe. This is an animal that was irradiated just for a short period, and then it has seizures because of neurological damage due to bilirubin toxicity, and this animal will die just in 1–2 days after the movie because of bilirubin toxicity. So, as I mentioned you before, also there were problems with the animals that were treated. The 20% survivors of the blue light up to P8, they show no important defects, so indicating that the main window for cerebellar development, just treatment up to P8 was sufficient for getting a quite normal structure of the cerebellum. But in any case, if we have done some more sophisticated analysis, this is a Golgi staining that specifically stained the dendritic arborization of the Purkinje cells, and we have seen that the dendritic arborization of these neurons was affected in these animals, that in any case they were affected on the survival. To map a bit better the window, we did the reverse experiment. So we put the animals under blue light after P8, that indicating, and then all those animals, most of those animals also died, just indicating that the main susceptibility window was between P8 and P12, more or less. So you can see here that there is an important difference in the depth of the cell layers of the cerebellum. Also, in the dendritic cell layer, you have much less dendritic cells, and the arborization of these cells is really very severely affected. So after having characterized more or less the bilirubin, the phototherapy effect, then we wanted to analyze also which were the other genes that could affect the phenotype. So in the clinics, it is well known that total bilirubin levels in plasma is not the only cause of disease, because in some cases there are many kids with very high levels of bilirubin that they don't have neurological damage, and there are many kids with much lower levels of bilirubin that gets affected. So this information clearly indicates that there are many other factors that participate in the severity of the disease, in bilirubin toxicity, and there are many candidates to that. Some of them are the modulator genes or some transporters that could theoretically could pump out bilirubin from the brain into circulation, protecting the cells. So two of those candidates are the MDR1 gene and the MRP, that both are genes that belong to the ABC transporter family. So the location of those genes is optimal just to pump out bilirubin from the brain into the cerebrospinal fluid or into the bloodstream. So to address this possibility, to have a better idea, because also in the literature there were contrasting experiments in cells, and results were not clear, so we did a genetic approach to determine more clearly what was the problem, which was the real role of these two transporters. I'm going to talk now only about MDR1, just to simplify the story. So we did double knockout. So we cross our Ugt1 strain with MDR1 knockout animals, and when we put those, we analyzed the survival of those animals without any extra treatment, and we have seen that there was no difference in the mortality. So both mortality curves were overlapping, and the bilirubin levels were exactly the same in both animals, indicating that the absence of MDR did not affect systemic bilirubin levels. But when just taking advantage of the experiments we did before with phototherapy, we put the animals in a condition in which mortality was borderline. So we had 50%, 60% of the animals that die, and this is the wild-type case I had already shown you in the first slide. And then in these conditions, all double knockout animals died, indicating that MDR1 has a very important role in the modulation of bilirubin neurotoxicity. And again, there was no change in bilirubin levels, just confirming that there is no modification in the systemic bilirubin levels in plasma. And what is also important is that bilirubin in the cerebellum triggers the activation of the MDR gene. So in those animals, we don't have MDR1 transporter that is normally activated upon bilirubin, high bilirubin levels. And this makes the animals more sensible to bilirubin toxicity. We also addressed a problem how to treat animals in an acute condition, or to mimic in a way what happens in an acute neonatal condition in babies, in which bilirubin peaks. So I mentioned you before that albumin is the main binder of bilirubin in plasma. So what normally happens is that bilirubin, unconjugated bilirubin, is bound to albumin, and then it is taken up by the liver. And then when Ugt is not present, then you have a lot of bilirubin that is not bound to albumin, because the buffer capacity of bilirubin is limited. And this is called free bilirubin. And this is lipid soluble, goes into tissues, and then goes to the brain and produces brain damage. Bilirubin goes also to other tissues that have a high content of lipids, but the brain is the only one that produces huge damage. So what happens normally is this, that in the tissue you have no bilirubin, because you have a lot of albumin and just a few unconjugated bilirubin, because you have the normal activity of the enzyme. In the case of hyperbilirubinemia, so you have extra bilirubin that goes into tissues, and the idea is that when you add albumin, what happens is that you add extra binding sites in the plasma, so bilirubin from the tissues gets just mobilized into the plasma, just reducing the amount of bilirubin, free bilirubin in the tissue, and limiting the damage. This is the strategy we try to do with our animals. So we did albumin infusions into our animals, starting at P1 every 24 or 48 hours. at different concentration, and this is what we observe. We observe that, well, this is the normal mortality curve of untreated mutant animals. As long as we increase the dose or we increase the frequency, then we arrive to save most of the animals from bilirubin toxicity. So animals that were injected every 24 hours with a dose of 5 grams per kg or 7.5 grams per kg, then they were fully rescued. And obviously the amount of albumin in plasma was highly increased. That was a dose-dependent concentration. Also the amount of bilirubin was increased in plasma, but it was enough just to free the tissues from bilirubin, and this is the correlation. You can see that there is a very good correlation between the amount of bilirubin in plasma and the amount of albumin that is in plasma. Obviously, we analyzed also the histology, and we have seen that the animals were fully normal because of the treatment, and the animals that did not survive, in which we transfused them with every 48 hours, then they had a very huge impairment in the cerebellum, and we also analyzed what happened between let's say the animals that were infused every 24 or 48 hours. We have seen that in the first 24 hours after the infusion, everything seems normal, but then there is a huge drop in the albumin levels in the animals after 48 hours of the injection, and this is the reason by which those animals are affected. And here also the free bilirubin, that is an indication of the amount of bilirubin that can produce brain damage. Then in the animal that is infused every 48 hours, this is very high. Instead, in the animals that are infused every 24 hours, the levels is really very low, and it is reflecting the histological and survival results. So you can see, well, I forgot this. You can see here the huge difference between the animals that were injected every 24 or 48 hours. So this indicates that this protocol can be, we expect that since there was no very clear data in the literature regarding the use of albumin in the patients, this is something that we think could be used later on in babies, just to postpone the moment of extra transfusion. That is not a very easy task, or just to avoid completely the extra transfusion by giving some extra time to the baby to trigger the expression of the endogenous Ugt1 enzyme. So also we show that the indicator of free bilirubin is the best indicator of neurological damage that is available. So the last approach that I'm going to tell you very briefly is a gene therapy approach. I mentioned before that the only available treatment for this patient is liver transplantation, which has a lot of problems, not related to mortality, but also to the availability of the organ, because there are not so many livers available for this type of disease, because physicians, many times they think that this is a disease that is not acute, so they can postpone the transplantation and give this liver to a person who really needs the transplant to survive, such as acute liver failure, hepatitis C, cirrhosis, in which if they don't receive a liver, they die. In this case, just they stay a bit longer under phototherapy, and they can survive. So we did an approach of gene therapy, different approaches. I think that you already know what is the AAV virus, adeno-associated virus, that is a vector that is used to carry, just as a cargo. to put your gene of interest into the genome of the vector, then to package the virus containing your cargo, and then to transduce animals using this virus to try to reinsert into the cell the missing or the mutant gene. So the first approach we did was using the Ugt1 cDNA and the CMV promoter, thinking that this could be just a good promoter because it was very highly expressed normally. So we injected animals at P2 just to give some time the animal to express the transgene. We kept the animals under phototherapy for some days, and then we removed the animals from phototherapy. We observed that all treated animals survived. This is the black continuous line. All animals survived, and they were absolutely normal. This is a rot test that also was mentioned before during the previous talks, in which you put the animals just to, is a test used to determine motor coordination. We have seen before that the main region of the brain affected in our animals was the cerebellum, which is related to motor coordination. And all animals were absolutely normal. Bilirubin levels were relatively low. You can see here that this is the untreated animals, and we go from here to here after 15 days, and this is after 150 days. So there is a rise in bilirubin levels over time. That this is something we try to address in the next approach. And then you can see here that histology of the treated animals is completely normal. So what was strange in our case, well, this virus was AAV serotype 9, that it can transduce different tissues, and liver, heart, diaphragm, skeletal muscle, they were positive. But what was strange is that we didn't find any expression in the liver, but we find expression in the skeletal muscle that was responsible for the protection of bilirubin toxicity. And well, this was due to a shutoff of the CMV promoter in the liver. And in any case, activity in the skeletal muscle was apparently sufficient to get resistance to bilirubin toxicity. Then we did a second approach that since the first one we were not satisfied with that one, because if we had first expression in extrahepatic tissue, that if you have to do that and convince a physician to go for a therapy doing what is not normally going on in physiology, then it is very complicated. So we use a liver-specific promoter, and you can see here that after, this is the mutant animal and this is the wild-type animal, after 2 days already this viral vector is expressed and can reduce significantly bilirubin levels up to the point that you cannot distinguish anymore which is the mutant and which is the wild-type animal. And you can see this here just with the total bilirubin levels analyzed just a few days after the transduction. So also here we analyzed over time. We had animals that were maintained alive for more than 17 months, and even if we had some increase in bilirubin levels, were really rather low and very, let's say, under the threshold of bilirubin neurotoxicity, well below this threshold. So albumin levels were the same exactly in wild-type and treated animals. They performed the same, and the histology of the liver was completely normal. So since we still had this loss of efficiency over time, then we did a third approach that was based especially in the injection, just the comparison between neonatal injection or neonatal plus adult or only adult injection. So we did that. Also thinking because it is known that after neonatal transduction of AAV virus, this is a virus in which the DNA remains episomal and it is lost very fast. You can see here different ways of determining the viral genomes that are remaining in the hepatocytes over time. You can see that after 1 month, there is a very huge decrease, and then it remains stable for longer time. And this is a real-time PCR of genomic DNA. This is Southern blot, and this is activity. All three ways of determining activity or genomes give the same result. This is 10% of activity of the normal wild-type levels. So when we did this type of approach, we have seen that in the case in which we did before, it is exactly the same as what I have shown you before, that it increases a bit over time. These animals had 13 months, more than 1 year. When we repeated the injection P4 plus adult at P60, using different serotypes to avoid the immune response against the capsid. So the efficiency was very, very good, but was even better when we transduce animals as adults. You can see here that the bilirubin levels were completely undistinguishable between the wild type, that is the blue, that are the blue points, or the adult animals that were transduced. So this approach was really very efficient. So we are now doing a different approach. This different approach is based on the optimization of the sequence of the Ugti cDNA. So we are using different strategies to optimize the gene that are based just in the codon usage, that is codon optimization, removal of different sequences just to avoid some proteins that could affect the stability and the translability of the mRNA, or to remove cryptic splice sites or sequences that could produce a secondary structure affecting translability. So this is our next approach. Well, this I will skip. Well, just I want to show just this point here is that in some cases this is a factor 8 that is the factor that is missing in hemophilia. And you can see here that after codon optimization, you have about 40 times increase in the efficiency of in the activity of the factor, and this is due to increased translability and stability of the mRNA and the protein. And this is just the last slide, in which I would like just to show you that we have tested different constructs. Not all of them work. Here we have different, what is called optimized one. This is even worse, apparently, than the wild type. And we have one that apparently works much better than the wild type that we are planning to use. In this respect, we are trying to do now, in collaboration with other groups from Europe, a clinical trial to using this construct just to transduce persons affected by Crigler-Najjar with AAV virus to try to correct the disease. So this is more or less all. So the conclusions, I think that we can go very fast. I don't know how much time I have. Finish. Okay. Just okay. Well, conclusions. I have said them over the seminar. So we think that this is a very good model just to reproduce the human disease because it mimics the most important features of the human syndrome, and could be very useful just as a preclinical model to understand the disease or to treat the animals and get better protocols to treat the human syndrome. So just I will skip this, and then I will go directly just to help to thank the people who was working in the experiments. I have to say that most experiments were done by Giulia Bortolucci, who is an excellent postdoc in my lab, and look at it, the part regarding MDR. Simone, the part of albumin. Fabiola is also here. She's doing also other experiments with those animals, and well, the rest just different groups in Europe, and that are collaborating with us for different aspects of the experiments and the funding we have just to support the research. So I am here for questions.
Thank you. So now we have questions to Andreas.